I am Sutapa Datta, present again in front of you to welcome in my channel Sutapa's Biology. In my last video, I have told you about a basic idea about the enzyme. Now today also I am starting the second part of the enzyme. So enzyme 2. My today's topic is again enzyme and second part of that. In the last video I have told you lastly about the types of the enzyme, that is the simple enzyme and conjugated enzyme. And now in this portion, in this part enzyme 2, first I shall discuss about the classification of enzyme. Classification of enzyme. Now what is classification? Classification of enzyme. Here we shall observe that there are 6 types of enzymes. Number 1 is that, number 1, oxidoreductase. Oxidoreductase. From the name it's clear that it's perform or catalyzes the oxidation and reduction reaction. Reductase. It can perform the oxidation as well as reduction. And example of this type of enzyme is cytochrome oxidase. Like that in respiration we can get the cytochrome oxidase. Now cytochrome oxidase is very important one which we can get here. The example is cytochrome oxidase. It has big role, it has great role in enzyme action. Later I am telling you about that. So example is cytochrome oxidase, dehydrogenase, etc. Number 2 is transferase. Transferase has the role to transfer the group from one to other. Like that we can say hexokinase. That's very important enzyme, very popular one, present in glycolysis. Transferase, hexokinase. Number 3 is that hydrolase. Hydrolase means the enzymes which perform the reactions or catalyzes the reaction in addition to water with the substrate. That is, in case of digestion, in every step we can see the function of the hydrolase. Like that, example is amylase, lipase, etc. These all are hydrolases. That means they can do the hydrolysis reaction. Hydrolysis, hydro means water. So hydrolysis reaction they catalyzes. That's why they are hydrolases. Number 4 is that lyases. Lyases means it also we can say that the mechanism of that to it just like that it will break the double bond like that. Like aldolase, example is aldolase. Number 5, I am erasing this word. Number 5 is ligases. Ligase is the enzyme which help to join the different reactants or products, etc. Like that DNA ligase, which is genetic glue. DNA ligase. It's the example. It's a genetic glue. And the last one is isomerase. Isomerase. It has only function in case of the isomers. I think you know about the isomers, isobers, etc. Now isomers, they have the, like, one example, very common example of isomer is glucose and fructose. They have the same empirical formula. They have the general formula is same, C6H12O6, but they have different structural formula. So this is about the isomer. Now what's the function of the isomer is? For the interconversion of the isomers. Interconversion of the isomers, that is forming the fructose from glucose or glucose from fructose, etc. So this is known as isomer, and here we can give the example of like that, that the triose phosphoisomerase. Triose phosphoisomerase, which is present in glycolysis. Triose phosphoisomerase, it can change the phosphoglyceraldehyde to dihydroxyacetone phosphate or dihydroxyacetone phosphate to phosphoglyceraldehyde. So this conversion can be done by the effect of the isomerase. So this is about the classification of the enzyme, and now we are coming to the mechanism of enzyme action. And some terminologies I shall still explain to you. That is, one is, what is activation energy? Number one terminology is activation energy. सुने हैं क्या आप लोग एक्टिवेशन एनर्जी? एक्टिव मतलब ही एनर्जी है, और एक्टिवेशन एनर्जी, व्हाट इज दैट? सो एक्टिवेशन एनर्जी, to initiate any reaction, chemical or biochemical, any reaction, there must be some energy which is required to initiate the reaction. Now this what's the relation of enzyme with this activation energy? So amount of energy which is required to start start a reaction, amount of energy which is required to start any reaction is known as activation energy. So this is the term with the which is related with the chemistry. Now you are thinking why I am telling this here. One thing is that the in enzyme is a topic in chemistry also, and another thing is that the activation energy, role of the enzyme in activation energy, that enzyme will decrease the amount of the activation energy. Suppose कोई चीज हम लोग 100 रुपया में खरीदते हैं, तभी कूपन मिला हम लोगों को 50 रुपीज ऑफ, सो उसमें हम लोगों को कितना 50 रुपया में ही वो मिल जाएगा, तो आधा ये अमाउंट में मिल गया. Like that, in case of the enzymatic reaction, the activation energy is that gives the activation energy, suppose it's the 100 kilocalorie for any reaction, but the enzyme, if enzyme is participating in that reaction, it will reduce that rate up to 50% or 40%, like that. Any amount it will decrease. So what's the role of the enzyme in activation energy? That enzyme will lower down the activation energy, and that's why it will help the organism naturally. It will help the organism to perform all the reactions in low amount of energy. Okay, so this is the activation energy and this is the role of the enzyme in activation of energy. Next is another terminology, there is the turnover number. Turnover number is, we can say that the enzyme कौन कितना efficient है, कौन कंपटीशन में टॉप में है, वो हम लोगों को पता चलेगा turnover number से. How it's related with the enzyme action, that the turnover number is the number of molecules of substrate will be converted to product in a unit time. By one molecule of enzyme. मतलब कौन एंजाइम एक सपोज 1 मिनट में, यूनिट टाइम मीन्स 1 मिनट में कितना मॉलिक्यूल्स को सबस्ट्रेट से प्रोडक्ट में कन्वर्ट कर सकते हैं, वही उसका एफिशिएंसी का पैरामीटर है, वही मेजरमेंट है. ओके, सो लाइक दैट हियर द topmost one is carbonic anhydrous that can convert the 360 molecules per minute, 360 lakhs molecules per minute, 360 lakh molecules carbonic anhydrous. Turnover number is 3 per minute, 360 lakhs per minute. And the slowest one is lysozyme. It's present in our tear, and it has the turnover number only 30 per minute. So what's the difference in between these two, topmost and lowest? Now the next thing is mechanism of enzyme action. Mechanism of enzyme action, कैसे enzyme काम करते हैं, किसके साथ काम करते हैं एंजाइम, सबस्ट्रेट के साथ काम करते हैं, और क्या बनता है, प्रोडक्ट बनता है. तो चलो स्टार्ट करते हैं मैकेनिज्म ऑफ एंजाइम एक्शन. होप यू आर एंजॉयिंग दिस चैप्टर. ये अच्छा चैप्टर है, मेरा तो बहुत पसंद का चैप्टर है, बहुत बड़ा भी नहीं है और अच्छा भी है, इंटरेस्टिंग है. सो मैकेनिज्म ऑफ एंजाइम एक्शन, फर्स्ट वी हैव टू नो अबाउट द टर्मिनोलॉजीज और डिफरेंट notations, not terminologies, rather to say the notations. Now here the first thing is substrate. That means the enzyme will act on it. This is denoted by S, capital S. Next, enzyme, that is the capital E, and then product, that is the capital P. So these three are the participants in the biochemical reaction. These three are the participants. It may be that the substrates are 1 or 2 or more than 2, but all are substrate in that case. And enzyme will be 1 generally, and product might be 1 to and more. Okay, now when it will act, what will be formed? Here I shall use the notations, not the full form. That is E plus S, or S plus E also we can say, but generally it's used that E plus S. Reversible, all the enzymatic actions are reversible, so this is also reversible. ES, that is the enzyme substrate complex. Enzyme substrate complex. Now this will be converted to EP. So this is ESC, we can say enzyme substrate complex, and this is EPC, that is enzyme product complex. Enzyme product complex, and ultimately it will form the enzyme plus product. Two differently enzyme plus product. Now this is about the this is the reaction equation of the reaction with the notations. Now I am giving the diagram, how it will look diagrammatically. I am giving the structure of enzyme in this manner. Suppose this is the enzyme. Now what's the role of this group? It's the active site. We have discussed in the last video that all enzymes has at least one active site. This is the active site. That time I have told you what's the function of the active site. That the active site has the function to bind the substrate, and the substrate is, in other words, it's known as ligand. Okay, so it will bind the substrate with it. Now we have to know about this, that this binding of the substrate will be in this manner, that the substrate should have to be at part with the active site of the enzyme. So this is the substrate, this is enzyme, and this is substrate. Now what's the need of this? Why the substrate should have to be the same at part with the shape of the active site of the enzyme? Actually, in active site the substrate will bind, the substrate or ligand will bind and will exchange the electrons, protons, and they will break the bonds, etc. Now for these chemical reactions they need the close proximation. If they are staying apart from each other, then how they will participate in reaction? So they have to be एकदम सटा हुआ रहता होगा, एकदम सटा हुआ रहेगा एंजाइम के साथ सबस्ट्रेट, तभी रिएक्शन होगा, otherwise there will be no reaction. So they have to make the complex in this manner that they will form the enzyme substrate complex, where they will stay very close to each other. And this is known as ESC, that is enzyme substrate complex. Now in the next one, here the enzyme and product will be separated. Now enzyme has no role for this reaction as product is the target and product बन चुका है, तो अभी क्या जरूरत है एंजाइम का? कुछ जरूरत नहीं है, तो एंजाइम को छोड़ देंगे. छोड़ देने के लिए ये देखो आप लोग, जो प्रोडक्ट का जो स्ट्रक्चर है वो एकदम डिफरेंट than the enzyme. Product will be like this. Suppose this is the product P. So it has no structural similarity with the active site of the enzyme. So naturally it will not stay in this active site and it will ultimately release the enzyme. So this is about the basic frame of the mechanism of enzyme action. Now we are going to the next thing, that is the models of the enzyme action, and that will be in my next video. Okay, so watch करते रहिए. हाँ, तो आप लोगों को सब कुछ मिल जाएगा इसमें. हाँ, और भी एक गुड न्यूज है जो इसके बाद हम लोग शुरू करेंगे जेनेटिक्स. आई थिंक यू हैव इंटरेस्ट इन जेनेटिक्स and we shall start genetics after this. That is after the enzyme, that will be a biomolecule where we shall study about the structure of the DNA, RNA, etc. Because in genetics उन लोगों का ही स्टोरी है, तो उन लोग कैसा है वो तो जानने का जरूरत है, तो वो पहले जानेंगे, उसके बाद हम लोग शुरू करेंगे जेनेटिक्स. So be happy, cheer up, bye for this video.